Dr. Pragya, please.
Good morning, respected judges and fellow colleagues. I'll be discussing my thesis on study of candidate gene polymorphism in patients of diabetic retinopathy in Indian population. I have no conflict of interest or financial disclosures to disclose. First, I'll come to why are we focusing on RAGE gene? It has been shown that the microvascular complications of diabetes at the sites, they have high age, advanced glycation end product formation. The age is attached to the RAGE gene which is coded by chromosome 6, a member of the immunoglobulin superfamily. This chronic activation of this RAGE gene has a role in the diabetic microvascular complications. Identifying genes that may be responsible for the severity may help in understanding may help us understand better the pathogenesis of diabetic retinopathy. The aim was to detect two polymorphisms, 374T replaced by A and 429 thymine replaced by cytosine in the single nucleotide polymorphisms in RAGE gene in healthy controls, diabetic patients with and without retinopathy. These were the two polymorphisms which were studied in the promoter region of the gene. It was a prospective case control study with 90 subjects, 30 healthy, 30 diabetics with retinopathy and 30 without. It was done in retina clinic at AMU. 10 ml of blood sample was collected following WHO precautions. 7 ml was kept for biochemical investigations and 3 ml was taken up for genetic analysis. First, these samples were first subjected to centrifugation and then stored in EDTA vacuotainers at minus 80 degrees Celsius till it was time to analyze them. Spin column based DNA isolation kit using the manufacturing instructions were used for DNA isolation, and when the DNA isolation was done, samples which had purity less than 1.7 to 1.9 were excluded from the study. The DNA samples were amplified using PCR, and specific restriction endonucleases ALO1 and TSP509I were included with specific incubation periods, and restriction fragment length polymorphism was actually used to determine the band length, and this was imaged using the BioRad software. These were the two bands that we achieved. The length of these bands was correlated with an image which was given by Barry Hudson et al. With the pioneer study in 2000, where he gave which polymorphism will correlate with the length of the band achieved. Results of my study: 374T thymine replaced by adenine polymorphism was significantly associated with patients of diabetic retinopathy with a p-value of 0.01. Of the examined polymorphisms, only the mutant allele, that is the adenine, was seen to correlate with the severity of diabetic retinopathy. We assumed this was all assuming the dominant model of inheritance. 429 thymine replaced by cytosine was not associated with diabetic retinopathy. When compared with healthy controls, the diabetics and the 374 T by A polymorphism was more in diabetics and diabetics without retinopathy. However, this correlation when done between diabetic without retinopathy and diabetic patients was not significant. However, the odds of patients having this polymorphism in diabetics with retinopathy was more. No such correlation was seen for 429 T by C. Duration of diabetes was a significant risk factor for development of diabetic retinopathy. HDL, blood glucose, fasting PP, serum creatinine was significantly associated with patients of diabetic retinopathy. In hemogram, RBC count and MCHC were significantly correlated. We believe this is an incidental finding. What is the relevance of our research to the existing clinical science? The existing results which are controversial with no common consensus within them. This is the second study from the Indian subcontinent with 4 other studies from the Chinese. There is no data available for the North Indian population. It is a pilot study which will pave way for a large-scale multicentric population-based study. Any alteration will help us devise rapid diagnostic kits and will become a target site for future CRISPR technology. So I'd like to conclude that 374 T by A polymorphism is significantly associated, while 429 T by C is not associated with polymorphism. Collaborative efforts are needed to develop an international and national consortium for diabetic retinopathy patients, and large-scale genome-wide association studies are the need of the hour to identify these. Thank you. The limitations of my study were serum RAGE levels. We have not correlated with them, which could help in determining what is the translational efficacy of this polymorphism. Any number of polymorphisms are associated. We have restricted ourselves only to RAGE gene polymorphism in the promoter region, and it could not be, this was a cross-sectional study. Effect on the progression has not been noted. Thank you.
Since you are seeing the gene to be more common in both diabetics with retinopathy and without retinopathy,
Yes,
as compared to controls,
Yes,
right? But there's no difference between both the diabetic
Yes,
groups. So how does this information help us?
Ma'am, the odds of this mutation occurring in the diabetic patients with retinopathy is more compared to those without retinopathy. However, if the sample size would have been more, then we could have, because the p-value that we found was 0.03, 0.3. So if the sample size and due to the funds restriction, we could not increase our sample size.
So, okay.
That
So
would
your
have been-
sample size was calculated to find a difference from controls or to find a difference between the two diabetic groups?
Ma'am, the aim was to between the two diabetic groups, but with in the given sample size,
You couldn't find
we
that
couldn't
difference.
find. No, ma'am.
And is this genetic difference that you noted in your subpopulation the same as what is reported in literature from
Yes,
other population ethnic groups?
Similar study was done with National Eye Institute in Arvind and SN in 1991. They had studied 13 genes and found this 374 T/A polymorphism to be significantly associated for the RAGE gene. But they had most genes because it was a large study with around 500 subjects. No, no, sir. It is mostly in the Southeast Asian subcontinent. Yes, sir. There are 5-6 studies from Chinese population. This is the second from Indian population, and there is one study they are doing in Pakistan right now. They have not, I think they are about to publish. No, sir, not in Western literature. No, sir. Sir, because China and India are the predominant areas, diabetic capitals of the world, so could be, and sir, large scale funding is needed for this.
Thank you so much.
Thank